Hello and welcome to the NGS Webinar Series hosted by the Next Generation Sequencing Competence Network. Along the series, you will be hearing from scientists and clinicians from various fields of research. However, they are all connected by sequencing. Who would have guessed? We bring you to journeys where researchers are telling us about their sequencing data harvesting and bioinformatics analysis and surely results and perspectives. How do they do it? What is it good for? Watch and let us know. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We'll leave the link in the footnotes, and here is QR code for your fast access. Welcome aboard. Find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
Oh, thanks for the introduction. So my name is Anna-Lena Börsch. I'm a PhD student in the research group of Professor Meyer-Hörste at the Clinic for Neurology at the University Hospital in Münster. And I'm happy to talk today about polyneuropathies and about their cellular complexity, where we used a multi-omics approach. First of all, I will give a short introduction into polyneuropathies because I think probably most of you are not really common with this disease. It is a disease of multiple peripheral nerves and is among the most common neurological diseases with a quite high prevalence of around 5% of the population. It's more common in males than in females and has a higher prevalence in the older population. Patients often suffer from progressive sensory and motor impairment with a high socioeconomic impact. And the underlying mechanisms or cause of PNPs are diverse. So there are multiple risk factors like alcohol abuse or toxic medications, vitamin deficiency, but also multiple chronic diseases as, for example, diabetes. PNPs are classified into different subtypes. For example, in diabetic polyneuropathies or in chronic inflammatory demyelinating polyneuropathies or in chronic idiopathic axonal polyneuropathies. There are also a lot of other subtypes of PNPs. I will not mention all of them at the moment, but probably later on. But the problem is that in most or in up to 30% of the etiology of PNPs remains unclear. And potential or a diagnostic potential is especially important to detect treatable causes such as immune-mediated neuropathies, which account for up to 10% of the polyneuropathies. I already told we have a peripheral nerve disease, so I will also give a short introduction into the peripheral nerve system and also the nerve structure. Peripheral nerves are those that connect the central nervous system with the neuromuscular junction of the limbs and the organs, and it includes the cranial nerves and the spinal nerves, and the roots and branches, peripheral nerves, and the neuromuscular junctions. And every nerve here, you can see a representative or an image of the nerve, has a specific structure. So we have the epineurium, which is composed mostly of collagenous extracellular matrix, which surrounds the entire nerve. Then we have the perineurium, which isolates a group of axon Schwann cells units to form a nerve vesicle. It constitutes the main diffuse barrier between the endoneurium and the extravascular tissue. So it's a nerve tissue barrier or a blood nerve barrier. Here. This is this one. And then we have the endoneurium, which is a layer delicate connective tissue around the myelin sheaths of each myelinating nerve fiber. And of course, we also have other cells in every nerve. So I already mentioned myelin sheaths, and they are surrounded by myelinating Schwann cells. Then we have like the axons, we have blood vessel, But we also have like resident macrophages or non-myelinating Schwann cells and fibroblasts in our nerves. Today I will present one of our studies, which was recently accepted by Nature Communication, so you can hopefully read our publication really soon. It was a study of 3 centers here in Germany, so Essen, Würzburg, and Münster in total. So we collected several nerves from in total 33 PNP patients and 4 controls, and performed single nuclei or isolated nuclei, and then performed single nuclei RNA sequencing and created a really large single nucleus transcriptomic atlas of the human cerebral nerve. Afterwards, we confirmed and also underlined our findings with different approaches, as for example histology, where we used RNA in situ hybridization and immunofluorescence, but also immunohistochemistry, and also spatial sequencing. And I hope with this talk I can convince you why a multi-omics approach is really helpful to study a disease like polyneuropathies. Yeah, first of all, I already told you we performed single nuclei RNA sequencing, and after removal of doublets, low quality cells, and also batch corrected, we ended up with around 370,000 total high quality nuclei cells and 24 individual clusters. Thereby, we have 5 clusters which are associated to the peripheral nerve sheath cells. So we have here the endoneurial cells, the perineurial cells, and the epineurial cells. And we were kind of surprised that we found 3 different populations of perineurial cells because it was not previously described in rodents that they are so heterogeneous. Then we found 4 different Schwann cells clusters. So we have the myelinating Schwann cells, the non-myelinating Schwann cells, and also Schwann cells with a damage and a repair phenotype. We found 8 different vascular cell clusters of vascular smooth muscle cells, pericytes, and also a lot of different endothelial cells, which form a continuum from a venous capillary gradient to arterial. And we also found some lymphatic endothelial cells. And we could also find in our data set 6 nerve-associated leukocytes cluster, which mainly were of myeloid lineage, so outnumbering the T cells and B cells. And the next step, so we had this 24 clusters in the end, and in the next step, we employed in situ amplification-based spatial transcriptomics with a subcellular resolution, so spatial seq. Here we use Xenium, and in total from 8 patients, and we designed a custom panel of 99 genes where we put in known marker genes and novel transcripts that we found in our single nuclei RNA sequencing data. We matched each cell from the spatial sequencing to a single nuclei RNA sequencing cluster and defined thereby 8 larger group, which we visualized in an FFPE cross-section of a human cerebral nerves. Here is shown a control. The nerve consisted of multiple vesicles formed by, which I outlined here in black, and each was formed by endoneurial cells. So here in red and in blue, you can see them lining in the middle. And they were surrounded by perineurial cells, which are here in green. And we also found vessels, which were outlined by endothelial cells and other vascular cells, like vascular smooth muscle cells here in orange and yellow. And we also found some epineurial cells here in purple, which were located in between of the vesicles and were also interspread with immune cells. So with this approach, we morphologically confirmed our unbiased annotation of human peripheral nerve cell clusters. In the next step, we systematically compared cell type markers in humans with their published rodent counterparts. And as kind of expected, the majority of transcripts were shared between species. But we found MILP RIG3, PRIMA1, and CXCL14, which seem to be novel markers in our human data set for specific cell types. And they were not or were undescribed in rodent and in human literature. So we plotted known marker genes of perineurial cells like Claudin-1 or SLC2A1 in our spatial sequencing data. And then we also plotted our new marker, CXCL14, and we really saw that it's co-localizing with the known marker in the perineurial area. But we went one step further, so we also wanted to see if we can see CXCL14 expression on a protein level. So we performed histology, immunofluorescence on a human tissue with CXCL14, and as well we were able to detect CXCL14 expression in the perineurium. And in the mouse, we performed RNR in situ hybridization, and we were also able to confirm there is a CXCL14 expression. So with this approach, we were able to describe CXCL14 as a novel perineurial cell marker in human and in mouse nerves. CXCL4 is the receptor of CXCL14, and it's, as you can see here, mainly expressed in B and T and NK cell clusters. And we performed afterwards a cell interaction analysis where we saw that where a predicted interaction of B cells via the CXCL14, CXCL4 axis was predicted. So immune cells may thus signal perineurial cells in human peripheral nerves. So I sum up my first part. We defined with single-nucleus RNA sequencing and spatial transcriptomic a baseline anatomy of the nerve. We identify novel marker and define the anatomy. What is our expectation? Because of course you always have a hypothesis how you went in a research question. And yeah, I already mentioned chronic inflammatory demyelinating PNP, and it's already in the name, like the demyelinization. And what is happening there? So we have a damage of the myelin sheath around the axon. And where's the axon located in our cells? So we first have the axon, then we have the myelin sheath around the axon directly, then the Schwann cells, and then the endoneurium. And then all the endoneuriums are surrounded by the perineurium, and then later from the epineurium. So what you would probably expect by a disease which is affecting like the myelin sheath, that you will mainly see a change within the endoneurium and the Schwann cells. What that would mean for our single nuclei data, we would probably expect like the biggest change in Schwann cells and in our endoneural cells. But yeah, just have us a look. So first of all, we did a check on the compositional changes in our single nuclei RNA sequencing data, where we compared PNP versus control. And kind of expected, we saw, or the most apparent change in PNP patient was the loss of myelinating Schwann cells and the reoccurrence of damaged Schwann cells and repair Schwann cells, which indicated a nerve damage, which we would probably expect, but we were kind of surprised that we also saw an increase of perineurial cells. And in the next step, we performed the correlation analysis with the INCAT score. It's the inflammatory neuropathy cause and treatment disability score. So it's a score which define like the severity of the disease. And everything what you see in red is positive correlated with disease, and everything which is blue is negative correlated. And also here we saw a positive correlated with disease of leukocytes, perineurial cells, and repair Schwann cells, and a negative correlation with myelinating Schwann cells. So immune and repair signature correlate with disease severity in polyneuropathies, but alter cell composition are also outside of the endoneurium. As we can see, perineurial cells seem to be changed. But of course, we also wanted to check on a transcriptional level. And here we performed a cluster-free neighborhood-based approach. And we saw that neighborhood with the most DE genes were located in vascular clusters and in the macrophage cluster 2 here, which surprised us at the beginning a bit because we were more thinking, okay, probably we will see the most changes in Schwann cells, for example. But we also performed a DEG analysis in selected nerve-associated cells, for example, myelinating Schwann cells. And we saw there an increase of fibrotic tissue remodeling, extracellular matrix formation, and immune regulation in polyneuropathies compared to controls. And also in repair Schwann cells, we saw an increase of nerve repair and neuronal differentiation. So PNPs induced a transcriptional change in Schwann cells, but also widely outside of the endoneurium, especially in non-endoneurial vascular cells. PNPs thus surprisingly affect the entire cellular micromilieu of peripheral nerves, even beyond the endoneurium, and that thus may constitute as a pan-nerve disease. So in the second step, we defined polyneuropathies probably as a pan-nerve disease, as we could see changes also outside of the endoneurium. Yeah, I already mentioned that we have a lot of different polyneuropathy subtypes. So we have our 33 patients, and we subdivided them into the subtypes of polyneuropathies. So in chronic idiopathic axonal polyneuropathies, in chronic inflammatory demyelinating polyneuropathy, in vasculitic neuropathies, and our controls, but also in non-inflammatory or other non-inflammatory polyneuropathies, diabetic polyneuropathies, cancer-associated polyneuropathies, and in other inflammatory polyneuropathies. But in my talk, or in the whole further analysis, we focused on only on this subtypes, which has more than 4 samples per group to do better statistics. Here we as well performed compensation changes between the different subtypes and control. And in all PNP subtypes, we saw a relative increase of damaged Schwann cells, repair Schwann cells, and the reduction of myelinating Schwann cells. So Schwann cells pathology thus occurred widely across polyneuropathies, but we also saw some specific changes for the subtypes. So, for example, for the vasculitis polyneuropathy and the chronic inflammatory demyelinating polyneuropathy, we saw an increase of perineurial cells and endothelial cells. We also performed spatial sequencing on the different subtypes. So I already mentioned that we did 8 patients in total, so 2 patients per condition. And also here we were able to confirm the loss of myelinating Schwann cells here in blue, and an increase of repair and repair endothelial cells here in green in vascular polyneuropathies, but we also saw it, but less prominent in CIDP and CIAD. Plotting perineurial markers in spatial sequencing images, we saw that we have a single layer in our control subset, while we have a multi-layered, multi-cell layered perineurium in PNP patients, especially in CIDP. And to investigate this in a bit more detail, we performed the EMA staining, so the epithelial membrane antigen staining, in 18 controls and 12 CIDP patients, and measured the area and the perimeter of the perineurium for each vesicle, so in total around 370. vesicles. And then we performed a linear mixed model to account for potential confounders like age and sex as fixed effects and random effects like center and sample-specific variability. And as you can see, we saw a significant increase of the area and the perimeter in CID patients compared to control. So CIDP exhibited focal perineurial hyperplasia driven by an expansion of PNP-specific perineurial cells. I'm not showing that, but we are kind of thinking of that it's potentially induced by immune perineurial CXCL14 signaling. So as a focal pathology, we saw a perineurial thickening in especially inflammatory polyneuropathies. I have already mentioned that we also found immune cells in our peripheral nerves, and we subclustered all of this immune cell nuclei. And as you can see, we found a lot of macrophages, but with this approach, we were able to also identify smaller subtypes like CDC2s, which were not described before. But also naive B cells and plasma cells and different T cells. But most of them were still myeloid lineages, of course, and mainly macrophages. In our spatial sequencing data, we quantified the leukocyte-associated transcripts like PGPRC, but also MS4A1 for B cells and CD3E for T cells. And we saw in all 3 PNP subtypes an increase in the density of endoneural and epineural leukocytes in all 3 subtypes compared to controls. In the next step, we performed compositional changes in the immune cell clusters. And surprisingly, we saw in all 3 subtypes an increase of this macrophage 18 clusters. So we wanted to know what is this macrophages 18 clusters. So we performed gene ontology term enrichment analysis of the marker genes of this cluster. And we saw that it was enriched in lipid metabolism related pathways. We also plotted marker genes known from lipid-associated macrophages, which are described, for example, in CNS myeloid populations in stroke, which have a phagocytic and lipid sensing phenotype. And we saw that they also seem to be specifically high expressed in the macrophage 18 cluster, why we called it now lipid-associated macrophages. Moreover, we projected stroke-associated myeloid cells, which we know from one of our other publications, which we published 2022, in our data set, and we could also see that they really align. And also when we stained marker genes here in red from our macrophage 18 cluster or our LAM cluster with BodyPy and myelin, not shown here, which labels neutral lipids, we saw that it's really co-localized. So macrophage 18 cluster does likely represent a population with a myelin phagocyting capacity similar to myelin debris clearing stroke-associated myeloid cells. And maybe it's shared between the central and the peripheral nervous system. Moreover, we saw that this macrophage publication expressed TRM2, and TRM2 is quite well known on disease-associated microglia known from Alzheimer's disease. And also when we quantified it in our spatial transcriptomic, so like the TRM2 transcript, we saw a clear increase in our vasculitis polyneuropathy, but also in CIDP and CIAP compared to our control. So probably one mechanism behind the polyneuropathies could be a lipid uptake by nerve macrophages. But of course, we also wanted to see if we can see differences between the subtypes. So here we performed again like this cluster-free neighborhood-based approach. And we saw, for example, for the vasculitis, polyneuropathies, that it was mainly a vascular inflammation focus. Quite similar in CIDP, so there we also saw more like a lot of the E genes in the vascular clusters, while in CIAP we saw like a more widespread pattern of the DE genes. So it seems that each PNP subtype thus induce a unique transcriptional and also composition response. But I think at this stage, our study is still limited to really say a diagnostic potential, because our sample size per cohort is still quite small. So that's why diagnostic potential is still with a question mark. Yeah, and with this, I already come to an end. So I will summarize everything again. So we analyzed human peripheral nerves in a 3-center-based approach. We had a quite big cohort of 33 PNP patients and performed a multi-omics approach with single-nucleus RNA sequencing, histology approaches, and also spatial sequencing. And we defined the nerve structure, the human nerve structure or dual nerve structure, and were able to identify novel markers for perineurial cells, non-myelinating Schwann cells, and myelinating Schwann cells. And we also detected this lipid-associated macrophages, which express TREM2. And moreover, we did a comparison between PNP and controls, and we found a loss of myelinating Schwann cells, an increase of repair Schwann cells, and of perineurial cells. So we detected a perineurial hyperplasia in PNP, and we also identified like this an increase of lipid-associated macrophages and leukocytes, and also of vascular cells in PNP. Yeah, and with this, I would like to thank you for your attention, but of course also all our cooperation partners, which I just mentioned here, but special thanks goes to Michael Heming and Jolene Wolbert from my group, who worked with me together on this project. And you probably already see here the Dresden Concept Genome Center. That's why I kind of entered the whole area of the next generation sequencing network here. And I'm happy to take questions. Also, if you have questions later on, you can just text me via mail, or I'm also on LinkedIn. And I'm happy to share that everything what I showed is also already published in a preprint. You can just check it out. Our data are available on a PNS atlas, and we are also happy to share our codes on GitHub. We are part of the competence network Peripheral Nerve, and we also created a consortium which is called LINK, Lipid Immune Neuropathy Consortium. And if you want to know more about our group, we are also on Twitter and our website. So I'm happy to take any questions.